We have backup, but we want Lisa to be able to be piped in right after this. So we'll see how this goes. Again, thank all of you for your patience with all of our technical challenges this morning with the AV. But thank you again for coming to the workshop. I'm really excited to present with my team on all of these kind of unique different approaches than the standard at OHBM, generally speaking, of using fMRI or EEG and other methods. So for this talk, I'm going to focus on the tomographic image reconstruction and all the things that are meant there. So learning objectives, mathematical basis for image reconstruction, the value of overlapping multi-distance FNIRS measurements, validation of image quality of the reconstructed data relative to fMRI, and the effect of computational imaging parameters. So with regards to image reconstruction, taking a step beyond what Chiara was talking about in the measurement-based approaches, we really want to focus on the distributed organization of brain function that especially this conference is really focused on relative to other kind of more optically oriented conferences. And that is, as we all know, various aspects of the function of the brain are spatially distributed across the entire head, and you can interrogate the brain function asking questions in a task-based manner, asking where in the brain lights up if you tap your fingers, or think about your grandmother, or you flicker a checkerboard at someone's eyes, or you try not to fall asleep during a talk like this, or you can ask about tests. You can ask about the brain function in terms of task-free approaches, used right now like resting state functional connectivity while someone is sitting there trying not to move and staying awake, or while someone is asleep, especially with little kids who are very challenging to get awake and engaged data from in an MRI scanner. Over the last 20 years or so, there's been a lot of work in these resting state data sets in terms of functional networks. And so the important thing to keep in mind is that disease, development, and aging all manifest as difference in these task-based systems or in these resting state networks. And so there are multiple opportunities for imaging and thereby, mapping our measurements of spatial temporal dynamics of brain function, going all the way from neuronal activity, which as we know occurs at the bandwidth of, milliseconds in very small neurons that are very hard to access in, people who are going to sign release forms, through local changes in calcium concentration, mitochondrial changes in cerebral metabolism, all the way up to changes in cerebral blood flow, and then into changes in local changes of oxygenation, or the blood oxygenation level dependent signal of fMRI that is akin to what we measure with functional near-infrared spectroscopy. So again, going all the way from very small, fast things that are very challenging to image in vivo in humans, up to very large and relatively slow things that are less challenging in humans. So there are a lot of different contrast mechanisms and a lot of different technologies that allow us to access these variances in brain function, from high-density DOT and FNIRS and fMRI that I'm going to focus on, diffuse correlation spectroscopy that Lisa will talk about next, positron emission tomography, and then, of course, magnetoelectrical potentials with EEG, which are, electrical leads on the scalp, ECOG, where you take off this skull and put the leads directly on the dura or penetrating into the gray matter of the head of the brain, and then MEG. So again, I'm going to focus on these three technologies for hemodynamics delay, and really want to again reinforce that imaging brain function with light isn't just fun and just a different thing to do than with fMRI, but it really addresses certain profound limitations of the fMRI technology. And so it just depends on what your scientific question is. We're not trying to in any way say that we're going to replace all of the glory of fMRI with NIRS, but there are very specific situations where fMRI simply just cannot go right now. For when people have contraindications due to having implanted metal of active electronics, for example. So the wearability is also important if you want to have freely engaged interactions in naturalistic environments, or portability in low resource settings, or at the clinical bedside, et cetera. Also cost. So for example, who here writes grants that have fMRI data? Anyone? This is the interactive portion of the workshop. So a few people. How expensive are MRI scans? Very. he said, laughing. Yeah, totally, right? So I'm going to go and take data from 100 people. That's many thousands of dollars with MRI, right? So when we're able to take data with NIRS, you have to buy the NIRS systems for a few $1,000s or whatever, but then you literally plug it into the wall, and then you can take hundreds of data sets, and basically the cost is for participant fees. They're not for facility maintenance. So you're able to increase your order of magnitude in the number of participants very, very readily for a given cost to your study. So again, the main focus that we have in our lab at WashU is to be able to provide minimally constrained imaging of spatially distributed brain function fully across the lifespan, with the perspective of having fMRI comparable image quality, but in form factors of systems that you kind of see here. On the top left is one of my twin daughters, Beatrice, wearing one of our kind of validation proof of principle fiber-based systems. Next to her is my friend Britt, wearing one of our large validation systems we published on in 2014. On the lower left is an infant that was undergoing cardiopulmonary bypass surgery. And we went and took about a dozen data sets in the surgery ward last winter. And just to highlight that even though there's kind of a lot going on there, you see all the fibers on the kid, you know, this was throughout a 6-hour surgery course. We're able to record semi-continuously throughout all of this without getting in any of the clinician's ways, right? So yes, you could go in there and do CT every now and then and revolve the X-ray around the participant. You can do EEG, which is way more sensitive to motion artifact than FNIRS is, but it really just highlights the opportunities to be able to look at cerebral hemodynamics and brain function, temporal correlation of functional networks in theaters that MRI just currently cannot get to. And then this is an image of Gower Labs, one of the few companies worldwide that are selling these fiber-less systems, these wearable systems. So this is kind of a concept design. They are selling some of these systems now, but this is the basic idea. I want to really highlight that this is a very active part of research in our community, and you know, the goal is to really get to something that's very wearable, very lightweight, and really opens the door to take fMRI comparable data out into the community. So just to touch again on what Hamid was saying in the beginning, you know, as a physicist, to first approximation, we're all walking spheres of salt water, right? And so if you want to measure the variability of something inside of a system, you have to probe it with energy. And so if we think about water absorption of photons, you're shooting energy in of a given wavelength, and you want to see how much of that energy can you get back out. So that's what this black curve is here. This is the absorption coefficient. The higher on this plot is, the faster into the tissue the energy is absorbed, and you can't get any information out. The lower parts are where you can probe deeply into tissue. And so again, in terms of like optical physics, deep is like a centimeter. You know, usually use optics to look at microscopy and things like that that are like microns, single to hundreds of microns deep. So if we look at very long wavelengths of electromagnetic radiation, the systems that generate those are fMRI machines. So the whole point is that we're trying to move beyond that technology. If you look at very high energy photons, you can get those through biological tissue. Problem is that they are ionizing, and so they can damage DNA, which isn't great for longitudinal studies in kids. So you'll notice that in the middle, there's this remarkable dip that's called the optical window, where optical and really near-infrared spectra wavelengths are able to penetrate multiple centimeters through living biological tissue. And that is what we are leveraging with functional near-infrared spectroscopy and diffuse optical tomography. So this very complex figure, as Emma showed earlier, just kind of shows the big picture perspective of all of our data processing. So I won't belabor anything too much. I just want to kind of jump off of what Emma was talking about, where if we focus in the lower left-hand corner here, the pre-processing pipeline takes you from data after simply taking the log mean step on the left-hand side here. After pre-processing, you can see a lot of the temporal patterns of the retinotopic paradigm that was repeated with a nice peak at the stimulus frequency after kind of removing the systemic signal. So there's still more to do between getting that level of temporal variability in our measurements across the head, again, on the system that Britt's wearing there, to get a map like this of tomographic brain function projected onto the cortex. I should say reconstructed in the cortex, not projected. So the first thing to keep in mind is that this is diffuse optical tomography. It's a model-based reconstruction technique, so you have to build a model that's appropriate to your question. So again, this is all utilizing examples from NeuroDot, this pipeline, or a set of toolboxes that we develop that are available on NITRC. And again, if you go there, you can download it and work through some of these tutorials as you're sitting there now if you want to. So to generate the head model, you have to start with anatomy. You can get that from an atlas if you don't have MRI, which is kind of the overall goal that we're all working towards. Or if you do have a set of grad students that you're going to run a validation experiment on, you have them do an MRI, you get their T1 and T2, you combine those together to generate a mask, which just means binary values that you label with scalp, skull, CSF, gray matter, and white matter. Why do you care about those? Because they all have different optical properties, as Hamid was talking about. So optical properties meaning the coefficient of scattering, meaning how often do photons bounce off of the tissue, and the coefficient of absorption, how often as a function of space or distance does that tissue then absorb that photon and then heat up the tissue. So from that, you can generate a three-dimensional finite element mesh to be able to solve differential equations in using Hamid's near-fast toolbox or other toolboxes that use Monte Carlo. All you need then is the location of the cap, so the array of sources and detectors. And we're developing photometric approaches so that you can do that readily. You can also use approaches by hand, but that's kind of beyond the scope of today's conversation. For the tissue segmentation, you can use FreeSurfer, SPM, FSL, near-fast slicer, or NeuroDot. I just want to highlight that NeuroDot was generated over the last decade or so very much from a perspective of being able to stand on the shoulders of the giants that developed image processing and image analysis routines in MRI, because you guys are decades ahead of the optical imaging field. So we want to figure out the optimal pre-processing for the optical data, put it in the same format of the fMRI data, and then we're off and running, and you can analyze data in NeuroDot or in any of the fMRI-focused toolboxes. So it's all the same, because we want to be able to have cross-modal analysis and comparisons, again, between DOT and fMRI. So importantly, you can make these models based on specific subjects, or you can have an age-appropriate atlas as well. For the mesh generation, you can use, not this HDMI connector again, whole head density mesh generation with near-fast or CGAO or isotomesh. Apologies, we'll see if this agrees with this again. And then again, intuitive and interactive cap placement. We have interactive GUIs in NeuroDot that you can find online. Let's see if this is going to behave. All right, then for the head modeling, once you have that, you want to solve the differential equations that Hamid mentioned at the beginning through that tissue. So you want to calculate what's called the Green's function. The Green's function, simply put, is you take a flashlight and you shine it into the head. The Green's function tells you where the photons are going to sample in the tissue. So for a source, it's just a fountain of photons. For a detector, it's just a vacuum of photons, but time reversed because of reciprocity in physics. So if you want to know what a source detector pair sampling looks like in the brain, where is it sensitive, you just multiply those two Green's functions together, roughly speaking, and that gives you something that is labeled there in part 3. So if you have a large array of sources and detectors, you can sum that sensitivity together to get your imaging domain, which we get by summing the A matrix there in part 4. And you can see that that's smooth, though it's only about 2 centimeters deep from the scalp surface in the head. But you can still look at how that intersects the cortical surface here in 5 and get a sense of the actual field of view that we have on a population study. And you can see differences in the colors. That's just because some people have big heads, some people have little heads, some people have differently shaped heads. So all of their light models are going to interact a little bit differently on the borders. but you can combine them all together and thereby look at where the data is variant across the intersection of all of those regions of interest, which, if you think about it, is actually identical to what's done in MRI, right? On the group-based data, there's small differences in the voxel field of view. So you combine all of that together, and then you're solving a linear set of equations, y equals ax, where y are your measurements on the surface, a is your sensitivity, generated as I just showed you here, and x are all of those variations, spatial temporal variations within the volume that you're trying to discover through this regularized reconstruction. And we'll talk more about that in the next few slides. So this is a movie of photon diffusion, a model of photon diffusion using Monte Carlo methods that I borrowed from our friend David Boas. Again, to recapitulate what Hamid said, you can model the fluence, which is just the photons going through an area per unit time. And if we're assuming small changes in the fluence driven by small changes in the underlying optical properties, so what that really means is that when you think, neurons in your brain fire. When they fire, they couple to the neurovascular coupling mechanisms and they ask for more blood flow, right? And so that change in neurovascular coupling leads to local changes in oxygenated and deoxygenated hemoglobin. Those are the two functional chromophores that change as a function of time dynamically. That's what directly leads to these changes in mu A. So absorption due to oxy or deoxyhemoglobin. So we look at our data differentially. So changes in the fluence are related to the negative log of our stationary fluence or our time trace of fluence divided by some baseline. So for our data, we usually use just the average of the data that you collected as the baseline, with the knowledge being that your brain, you're not, unless you had a stroke and blood is collecting in part of your brain, in part of your head, that like if you look at the fMRI data also, time traces go up and down around a reasonably defined medium. So that medium is what phi naught is. So you can relate differential changes in light level to, again, differential changes in these green functions times these green functions, which are stationary, times differential changes in the mu A, again, local changes of oxygenated and deoxygenated hemoglobin. Here again are the Green's functions for the source and detector. You multiply them together, you get this nice sensitivity banana for a specific source detector pair. So again, we can do that pencil and paper math with the German S. You can also do it with the sigmoid or the computer math to then again do y equals ax. I don't have to belabor all of this. So basically, again, y again are the source detector measurements on the boundary, x are the internal distribution of perturbations and absorption, and a again is the model. It's the sensitivity of the system, defined most strongly by the shape of the head, secondly by the internal distribution of optical properties, the speed of light, coefficient of absorption and scattering, and the location of your sources and detectors. And of course, we model our photon diffusion with near-fast. When we generate a model, we do so for a participant, given where their cap is. We run near-fast once, and it takes a few minutes, and then we have A, and we can reuse A, our sensitivity, all the time. You don't have to rerun this model every single time you take data. So it's a little bit of a computationally intensive part to form A, but once you form A, you're good to go for a long amount of time. So the important part, again, is inverting the model, because as Hamid said, you have a lot more voxels in X than you have measurements in Y, so we use the Moore-Penrose generalized inverse, as you do, to reconstruct and invert that matrix. And there are two parameters to do that for regularization that we'll discuss later. But basically, you can tune this one parameter, lambda, which is a Tikhonov regularization, to kind of control the balance between image noise and resolution. You can soften lambda and have it be a large value, and that gives you big, blobby, looking reconstructions that look a lot like projecting EEG into the brain. And you can get small values of lambda, pardon me, maybe. Apologies again. Small values of lambda will give you very high resolution image quality if the HDMI works. This reminds me of running auditory stimulus to people in fMRI. Maybe it'll work, maybe it'll work. Come on. All right, so optical neuroimaging utilizes functional near-infrared spectroscopy. Again, you have a source of light, you have a vacuum of light, and you have your measurement. If a source detector pair is close together, your measurement samples superficial tissue, just like Emma was talking about. If they are further apart, it images deep tissue, but now you get partial volume effects, which I know is a thing that the fMRI community is very familiar with. So variance in that source detector pair reflects variance everywhere along that sensitivity banana. So cool, we have brain function, but now it's brain function over this huge swath of the volume. So the trick is to combine multiple overlapping measurement pairs to be able to reconstruct good imaging resolution throughout your imaging domain. So again, functional, when the brain thinks, it changes color dynamically, as Chiara showed you. We can detect this blushing of the brain with near-infrared light. Spectroscopy, because we use multiple wavelengths, as both Chiara, Hamid, and Emma were mentioning, to recover multiple different absorbers, oxy, deoxy, and total hemoglobin. And again, multiple overlapping measurements reconstructed gives us an even imaging domain at the level of the cortex within the volume. So all right, that's enough math for now. The value of overlapping multi-distance nears measurements, as Emma had walked through, basically you can measure variance in the brain with the source detector pair. But if you're trying to map and create an image, then you want to use multiple measurement pairs. The field has evolved over the decades to go from single source detector pairs in the early 2000s, in the 90s really, to multiple source detector pairs, as you do with EEG, to more still that are close together. You can basically interpolate and do topography. Once you get overlapping measurement pairs, you can do tomography. Once you get multiple overlapping pairs that are multi-distances, that's where you get high-density diffuse optical tomography, which is what we're focusing on. There's lots of juicy details in these studies, and again, there are significant challenges, as Emma highlighted, that the signals occur at depth, and you have to always image through that superficial layer. So you have to be able to regress the data out of that superficial layer, as Emma explained very nicely. Just to really belabor the point for a second, you can run simulations. All of this can be done in the Neurobot toolbox for those of you that are into running simulations on different data sets. You can simulate different grid designs, a high-density grid, a squared grid, or a triangular grid, as are very often used in the field. And so what we're looking at with the point spread function is you can imagine having a perturbation somewhere in the volume. You just want to look at what that perturbation looks like after you reconstruct it. This is done in microscopy system design all the time. And so the point is that what you want is a very even, well-defined point spread function. So you see the high-density grid gives you a nice circular point spread function, whereas the square and the triangular grid, there's these like weird image artifacts that have characteristic geometries. Now, you can quantify metrics of these point spread functions, such as the localization error, which is basically where is the center of mass of your reconstruction versus where it actually came from. And you see that the high-density grid has very low error that is very even across the imaging domain, whereas these other F-nears grids, the localization error depends where under the grid you are, which means there's a strong spatial bias in your image reconstructions from these sparse grids. Effective resolution, so how big is the point spread function? Again, when you have a high-density grid, it's more even, and when you have a sparse grid, there's a lot of bias in the field of view that can lead to false positives and false negatives in group studies. And this is also shown using extended perturbations. So that's all in simulation. This is then an example using the same paradigm that we've been talking about, a flickering checkerboard. Has anyone done retinotopy themselves and understands how very annoying this is to stare at? Anyone? Everyone in my lab? Awesome. I'll see you guys. Very good. So this is an amazingly great stimulus paradigm to use for validation, though, because when you flicker back and forth at 8 hertz, A, it's awesome to look at, but B, it gives you a really strong signal-to-noise back in visual cortex. So this is one participant, 10 minutes of data, averaged across 8 blocks. Just looking at a visual flickering checkerboard in the lower left visual quadrant gives you a nice, strong activation in the back right visual cortex with fMRI. With our high-density DOT, the same person a week later, you get a very similar, not exactly the same, but a very similar reconstruction to the same data. Now, with the sparse grid, this is simply a resampling of the identical data. Same system, same electronics, everything. This is all about measurement sampling. Same green lines, again, same system. It's just that without all of the overlapping measurements, you're not as able to better triangulate the variance within the volume. This is exactly the same thing as like measuring earthquakes from just 2 seismographs to many, many, many placed over the surface of the Earth. The more measurements you have of the surface, the better your reconstruction of the variance within the volume is going to be. You can still measure it, but you might not be able to locate where it's going to be. So there are a lot of instrumentation challenges, which is why there are only a handful of companies worldwide that are making strong inroads with this. But basically, to get the high-density array to work, you have to have a detector that is extremely linear in its output over at least 6 orders of magnitude in light level, between about 13 millimeter closest nearest neighbor out to about 50 millimeter farthest nearest neighbor. But then you have to have another 2 orders of magnitude of signal to noise, because variations in brain function are about 1% of the total variation of any measurement that you take. Turns out that that's true with your fMRI data as well, right? If you just look at your raw bold time traces, the interesting variation that you see for brain function is always going to be around 1 to 10%. 10% for like retinotopy, which again is like a sledgehammer to your brain. If you ask someone, think about your grandmother, or doing a population-based study with resting state, even more so, you're looking at like 1% signal changes. So it's the exact same thing is true here with optical data. But that just enforces, again, you need like 7 orders of magnitude in these detectors, which is a very, very strong optoelectronics challenge, using avalanche photodiodes or photon multiplier tubes or silicon photodiodes that are done nowadays. Anyway, it's a very active and exciting part of what's going on in a few labs worldwide. So that's dynamic range. Crosstalk, being able to get all of those detectors and sources to talk to each other without confusing each other, is a very interesting electrical and systems engineering challenge that is beyond the scope of this talk, but I'm very happy to talk to you guys if you're curious about those details. Also, optical coupling at the scalp. If we look up here in figure C, those are just examples of fiber optic probes that are able to move in and out of a sliding kind of a top hat infrastructure, such that the fiber optics are able to engage with your scalp with local variations, because everyone's scalps are actually a lot more bumpy than you realize. Also, local variations in curvature, etc. You want to make sure that your cap is able to fit not just you, but also you and everyone up here. And that's a remarkable challenge. It's not just like we have a single bore that works for everybody. It's more like if you have a 64-channel head coil and expect everyone's head to fit inside of it, depending on your study, that might not always work so well. So we, in our lab, have developed multiple different caps to be able to image data from neonates within the first day of life throughout early childhood development into adulthood, based on variations of the same theme up here. And Chiara also went through many examples of other cap designs that are used throughout the community. So now, validation of image quality. So you know that it's not just fun, we actually are able to show that this all works. So again, a big focus in our lab is to stand on the shoulders of the giants that have already come before us, that are like shining the light on the pathway of making sure that technology works. So similarly with PET, and then with fMRI, retinotopy is an amazing way of validating that the technology is giving you the maps in the brain that you expect. So as was shown earlier, this is a flickering checkerboard stimulus, a cartoon of which is shown over there, but this is the data sped up a little bit. It's 8 repetitions of the stimulus for both the DOT in the middle and the fMRI on the right-hand side. 1 subject, 8 blocks. non-concurrently recorded data. That head modeling that I showed you earlier allows us to have voxel to voxel or cortical ridge to cortical ridge level correspondence between the two modalities, even though it's not simultaneously recorded. So with these data, we were able to show with a Brian White et al. Paper that the image resolution is about 1.5 centimeters with DOT, which is far better than sparse NIRS by itself. It's more about 3 centimeters is the resolution. So with DOT, we're able to push beyond that. And then the accuracy, so like the center of mass of your activation with DOT is within about 5 millimeters. And so that's good because that puts us within the range of about a gyral ridge for where these activations are going to be. Not to the level of fMRI again, but far better than EEG. So in terms of being kind of a middle ground technology that's useful. Maybe. So as Emma talked about, for that movie on the top left, you can place a region of interest in one of the voxels and get a time trace that you see there, oxy-deoxy total hemoglobin in red, blue, and green, if you care about imaging spectroscopy, and the Fourier transform of those are down on the bottom. Again, with the head modeling, we get the bold data exactly aligned in time and in space, showing how the bold data tracks remarkably closely the optical data. Again, non-concurrently recorded data. You can do phase-encoded retinotopy following Dr. Serino and many others. So we're just looking at maps of the phase, temporal phase, and spatial phase are the same thing with retinotopy, right? So if you follow the checkerboard moving around in the circle, you can just color that circle based on the pinwheel on the top part of the slide there. So time in terms of the polar angle phase reflects space in terms of the visual field, and you see that these pinwheel diagrams, again, between DOT and fMRI show pretty good spatial correspondence. The next level of validation we turned to was language processing distributed throughout the superficial cortex. Our friend Steve Peterson et al. Published this lovely paper in using PET data, what was the first imaging representation of the hierarchical nature of language processing, with hearing words and auditory cortex, seeing words and visual cortex, speaking words and motor cortex, and thinking about generating words in left lateralized dorsolateral prefrontal cortex, classically Broca's area, even though we can all argue about whether or not that distinction makes sense now. So anyway, this is data averaged across 5 people with DOT and also 5 people with fMRI. Again, hearing words, like you listening to me talk now, strong activations in bilateral auditory cortex. Reading words, 1 word per second, 15 seconds on, 15 seconds off. The GLM analysis shows strong activations in visual cortex. If you ask someone to, rather than just read it passively, but to imagine themselves saying it out loud, you then subtract from that map the map due to just reading it, and you get activations in bilateral motor cortex for like the motor mouth area. I still love that this is an actual thing that you can see with DOT or with fMRI, even though no one is like verbally doing anything or physically doing anything, just thinking about it activates motor cortex. If you play the same game, but instead of asking someone to imagine saying the word, but to imagine saying a verb associated with it. So if a noun is dog, you might think bark or kick or something like that. And when you do that, and again, you subtract from that map, the map for Imagine Speaking, you get this really nice localized map in left lateralized dorsolateral prefrontal cortex. I want to really highlight here that the cool thing is not just that the on activations are there, but you also have lots of built-in negative controls throughout your field of view, right? Rather than having a spatially focused pad, having a pad that covers the, a large amount of the brain gives you lots of positive and negative controls on the way. So same 5 subjects, non-concurrent data, fMRI, and then the overlap between the two. You can see that while there is some spatial variability in some things, that the agreement between the two modalities is very strong. And so again, this was published about 8 years ago now. Because we're optical people, we care about oxy as well as deoxy and total hemoglobin, so you can make spatial maps of all 3 of these contrasts. And variability in these maps are especially interesting and important when you're looking at variability in neurovascular coupling as maybe caused by things happening due to a stroke or changes during ECMO or surgical procedures, et cetera. So now, resting state, someone sitting in the cap doing nothing for 10 minutes other than staring at a cross and trying not to move, which is actually highly not nothing. So we can argue about that too, and with the HDMI cable. So if you plot a region of interest in visual cortex, you get a time trace. A region of interest in a homologous region, so visual cortex in the other hemisphere, a similar time trace with a reasonable correlation coefficient. Another region of interest in a functionally distinct region, so like motor cortex, and you get very low correlation. So as I'm sure all of you are familiar with at this point, mapping resting state networks with fMRI has been a thing since 2005 or so, and is highly ongoing in its use in the community, and parcellations, et cetera. And so, as I was talking about before, this is a much stronger benchmark for image quality than just task-based data, because you're looking at 1% changes in the data. You can go and collect more data, but you can't block average yourself out of having terrible SNR. So these are just spatial maps of correlation with visual, auditory, dorsal attention and area MT, which is fairly deep for optical, and a default mode seed. We are not able to see PCC with optical methods because it's really deep. So that's everyone's favorite seed, seemingly, for the default mode network. But there are other superficial nodes of the default mode network. This is just choosing one to appear in dorsolateral prefrontal cortex. And you can see that as we progress from sensory motor into higher order cognitive networks, that you get not only bilateral, but also anterior, posterior constellation of regions that are also recapitulated in fMRI. This is 10 minutes of resting state data without any scrubbing for motion or anything. Again, showing pretty good correspondence between DOT and fMRI. You can do this in infants as well. This is older work showing a baby optimized cap so that they can lay there in the ICU. The cap is based on neoprene that's very soft so that the infants can have it on their heads, in this case for up to an hour, with similar data quality assessments that Emma was talking about earlier. Similar light models, as I was talking about earlier, based on participant-specific, patient-specific, or atlas-specific MRI to generate the mesh, to generate the light model with where the array is. This is all work that was done by Silvina Faridol, who's now at Indiana University. So the cool thing I want to highlight here is even with this very limited field of view cap, she was able to distinguish between visual, middle temporal, so again, dorsal attention, broadly speaking, and auditory regions of interest, and generate these spatial maps of temporal correlation. And comparing that, this is, I believe, 8 babies, showing that the spatial maps with fMRI and DOT are, again, very similar to each other and are differentiable between these different regions of interest. Interestingly, the visual network has the highest bilateral connectivity relative to area MT, relative to auditory cortex, and that the DOT follows the bold level of correlation. Further, that makes sense given known changes in early life developmental changes in functional connectivity, both in humans as well as in mice and rats and whatnot, where visual correlation is very high, followed by auditory, and then followed still by other like higher order regions of the brain that take longer to develop and mature. So now, general learning objectives of what I've talked about so far, the mathematical basis for image reconstruction, because everyone loves calculus on 11 in the morning on a Sunday. The tissue and array model-based reconstruction we discussed, the value of overlapping multi-distance measurements. The more measurements, the better the high fidelity maps you're going to get. Though that leads to significant challenges in system design and putting the cap on. So real-time metrics of data quality are essential to really make sure you're getting good data. Akin to anyone that's using FIRM, that's becoming very popular in the resting state fMRI community. Third, validation of image quality of reconstructed data. Again, the optically measured brain function can have fMRI comparable image quality across multiple functional domains, even when recorded at the point of care and in naturalistic settings. So the last thing I'm going to talk about here is the effect of computational imaging parameters. So as with the HDMI nonsense this morning, thank you guys for all of your understanding and flexibility with OHBM and the challenges of doing an interactive course with COVID, moving in and out of the situation, and then back into the situation. So there are tutorials that, again, if you go to NITRC and download NeuroDot, there are tutorials for image reconstruction that kind of walk you through all the steps. There are the PowerPoints that this has a few slides from that have associated scripts in MATLAB that are commented that kind of help navigate you through those. And if you have any questions, my email is in there, as are Emma and Ari and other members of the development team, if you have any questions about any of this. So again, this tutorial walks through taking the pre-processing data through the light model into the reconstruction and shows a lot of visualizations of single subject data. I had a PO from one of the institutes at NIH asked recently in a JIT component of submission, for those of you that have to deal with all that, she said, are you able to get single subject data with NIRS? We've heard that you have to get group data. And I said, dear God, no, that's not a problem. As with fMRI, you can get single subject SNR that's very good with optical if you know what you're doing, and that's very important for people to know. So all of the data here is all single subject, single run, so you can really get in there and analyze the single subject SNR and play around with image processing and pre-processing parameters and kind of look at their effects on everything. So again, the pre-processing, superficial syndrome regression, et cetera, light model, reconstruction, spatial normalization, all different pipelines that we have tutorials for, as well as some that put them all together so that you can learn kind of how to do it all, and then also set up batch processing that utilizes XNAT, as Emma was talking about. So again, the pre-processing, you can see the before and the after with the other tutorial. Then this tutorial focuses on reconstruction and spectroscopy, loading in a sensitivity matrix, inverting that sensitivity matrix based on regularization parameters, smoothing that sensitivity matrix, just like fMRI data has to be slightly smooth, this data has to be slightly smooth as well. Spectroscopy and image reconstruction again. We have lots of data visualization tools and are developing more every month for both the raw light level data, like Chiara was talking about, as well as at the image reconstruction data. So this is a screenshot of something we call plot slices time trace that shows 3 views of the reconstructed data projected onto either subject-specific or atlas-based anatomy, along with the voxel-based time course. And so you can interactively click around any of those 4 panes, and it will either move the spatial maps based on where in time you care about. You can do this with unblock average data or block average data. So you can really explore the data at a lot of levels. You can also click around the volumes and look at how the time traces shift, or click around, sagittal, coronal, and transverse slices of all the data. It's a really, really lovely data visualization and data exploration tool. So as we were talking about before, you can look at image reconstruction from the perspective of oxygenated hemoglobin, which we usually focus on because that has the highest signal to noise for reasons having to do with neurovascular coupling. Just the changes in the oxy signal are larger by about a factor of 3 on average than deoxy, and by a factor of about 3 to 2 than total hemoglobin, or changes in cerebral blood flow itself. But you can look at underlying changes in mu-A if that's your thing. 750 has a very high absorbance relative to 850, so usually the data at 750 doesn't look like it makes sense, but the data at 850 often looks very similar to the data at oxyhemoglobin. If you look at oxy, deoxy, and total hemoglobin, again, after combining 750 and 850, you are able to look at each of those chromophores very specifically, and you can click around and look at different thresholding of the data, different, again, regularization parameters of the data to see how they interact. Third, you can tune lambda 1, which is the Tikhonov regularization, which we generally always use as 0.01, but I want to really highlight that that is a tuned parameter based on matching our system with this level of high density grid against participant matched fMRI data. If you have a different system with a different number of measurements, your regularization parameter will change because it is a function of the system itself. 0.01 is generally good in the ballpark for any system design, but again, if you have a very an even more high-density system with really, really, really good optoelectronics, you can make lambda 1 smaller and get higher resolution, but you have to be careful to not have system noise contaminate everything. Lambda 2 is called spatially variant regularization, and that helps us tune in how deep the reconstruction pushes the data so that we have good correspondence with fMRI. So you can play around with different parameters for that. Again, I apologize for the HDMI thing going on here. So again, we've covered the mathematical basis for image reconstruction, the value of overlapping multi-distance nearest measurements, validation of image quality of reconstructed data against MRI, and a little bit of the effect of computational imaging parameters. Again, Tikhonov regularization balances signal to noise, and spatially variant regularization, again, is a system-driven parameter that properly locates in space your image variance. So that's something that like you tune once for a given system design, and then it generally works on every participant. But again, because similar to when you use fMRI, there are people that partner with Siemens or GE or Philips or whatever MRI and tune all these things behind the scenes. So by the time the data is in FSL or SPM or whatever you're using, these things are already settled. So this is all kind of like showing you how the sausage is made so that you can tune these parameters yourself if you're interested in the optical processing of the data. But it's not like it's any different than fMRI. Trust me, looking at fMRI data for a long time. So with all of that, thank you so much for your time. All of this is based on really optimizing from adults to school-age kids to infants, and then including in infants in the neonatal ward. So hopefully we'll get this HDMI cable to work for Lisa, who's going to come at all of us from Spain. So again, thank you for your time, and give us just a minute to transition.